AS Level Biology 9700, Paper 2, Variant 2, February March 2026. And in this video, we will discuss question number 5 and question number 6. And question number 5 is DNA replication must occur in cells before mitosis can begin. So describe the role of the enzyme DNA polymerase in DNA replication. So here I would like to go in detail about this question. That is, the DNA polymerase helps in complementary base pairing. So it links pre-activated nucleotides to the exposed bases on the template strand via complementary base pairing as adenine to thymine and cytosine to guanine. And then it also helps in the formation of phosphodiester bond formation. So it catalyzes the formation of these phosphodiester bonds. So between adjacent nucleotides via condensation reactions, it constructing the sugar phosphate backbone. So it also helps in synthesizing the new strands exclusively in a 5 to 3 direction by adding new nucleotides specifically to the 3 OH group of the growing strand. So it performs a proofreading function to detect and correct mismatched base pairing. So in this way, I would like to write this answer on the given space as it links pre-activated nucleotide to the exposed bases on the template strand via complementary base pairing. Secondly, it catalyzes the formation of phosphodiester bond between the nucleotide molecules. And thirdly, it synthesizing the synthesizes the new strand exclusively in a 5 and 2 free end direction. So now the next question is, the two strands of a DNA molecule are described as anti-parallel. Explain the consequences of the anti-parallel arrangement of the DNA strands for the replication of DNA. You know that uh, we always say that the DNA molecules are described as anti-parallel. So DNA polymerase has an active site with a highly specific shape that is only complementary to the 3 end of the growing strand, that is 3 to 5. But discontinuous synthesis. So one strand, the leading strand, is synthesized continuously towards the replication fork. The opposite strand, the lagging strand, must be synthesized discontinuously away from the replication fork, resulting in short fragments known as Okazaki fragments. So in this way, uh, I would like to say that this process takes place. That is these two strands of DNA act like that. Then the DNA polymerase can add nucleotides only in the 5 to n direction. So therefore, one new strand is synthesized continuously while the other is synthesized discontinuously. So we can write these points in this way. These 2 points I would like to write over here. That is the number one is DNA race synthesize DNA only in 5 and 2. We have also mentioned it in previous question, that is 5 to 3n direction. So 5 to 3n direction. So this can also be the part of this. And second point can stand in this way, that is one strand is made continuously, which is called as leading strand. And the other strand discontinuously as okay exactly fragments by DNA ligase. So it was part B2 of the A. Now the next is part B, and part B is after the separation of complementary strands of DNA during DNA replication, the separated strands of DNA can become twisted and coiled together to form tangles. The enzyme DNA topoisomerase alpha 2 alpha is able to remove these tangles in a two-step process. Step 1, TOP2A forms a complex with the tangled DNA and catalyzes the cleavage cutting of both strands to allow the strands to separate from one another. And step 2, TOP2A catalyzes the repairs of the cuts made in the DNA so that the strands are complete. Etoposide is a chemical that can bind the TOP to a DNA complex formed in step 1. This makes the complex very stable and prevents step 2 from occurring. Suggest and explain why etoposide can be used to treat some types of tumor in humans. So, you know that the tumor cells are characterized by rapid unchecked cell division for mitosis to occur. So the cell must successfully replicate its entire genome during the S phase of the interphase. So by stabilizing the top 2A DNA complex and preventing the repair of the cleavage strands, etoposide introduces permanent double strand breaks into the DNA. So when a rapidly dividing cell attempts to replicate or separate broken DNA, the replication machinery stalls. Cells have internal checkpoints that detect severe DNA damage. So when the damage cannot be repaired, it signals the cell to undergo apoptosis, which is called as the programmed cell death. So effectively, it's shrinking or stopping the growth of the tumor. So these are the points we need to write over here. In short, briefly, I would like to write all this. Tumor cells undergo rapid, uncontrolled mitosis or cell division etoposide leaves the DNA strands permanently broken. Or fragmented because step 2 cannot occur. It prevents DNA replication from being completed. And this accumulation of DNA damage relation of DNA damage triggers cell death. So it was all about question number 5. Now question number 6 is there. Question number 6, just like fill in the blanks, we have to complete the given paragraph. So here you can see that a cell surface membrane consists of many different molecular components. The passage outlines the arrangement of some of the molecule components in a cell surface membrane. Complete the passage by using the most appropriate scientific terms. So the terms which can be used over here, some proteins in a cell surface membrane are located on one side of the membrane. Other proteins span the entire width of the membrane. Some of these are transport proteins and include proteins that are needed for facilitated diffusion are the channel proteins. So the word channel will be used over here. So these are the channel proteins that are needed for facilitated diffusion. So the fluidity of the membrane is regulated by the molecule cholesterol. So which interacts with the fatty acids tails of phospholipids. Interactions between this molecule and fatty acid tails are known as known as hydrophobic. Hydrophobic. Hydrophobic interactions. Because the fatty acids had play a hydrophilic role and hydrophobic interactions are between the fatty acid tails. So it was all about the question number 5 and 6, and in this way we have completed this paper. So please like, share, and subscribe my channel to watch more videos on other papers. So thanks for watching.